In fact, yeah, the microbiome is an indicator of the plant health and nutrient acquisition of grapevine plants. If you have a switch in the microbiome, for example, from the soil, from the roots of grapevine plants interacting with microbes, if you know before that the switch is occurring, or you will have more presence of pathogen, for example, you can make preventive measures, but not only based on detection of one microbe, but about the community. What you're offering with a WineSeq service, for example. And these farmers, the wine farmers, can have a vision of the fingerprint of their terroir, why it is unique, but also what can occur later on, and to make preventive solution before that the disease arrive, and also to preserve the good part of the microbiome, the good biocontrol agent naturally present with grapevine plants. By managing the vineyard, by making some cultural practice according to this, what you show in the WineSeq service, for example, when you detect such kind of pathogens with the abundance of this pathogen in correlation with the whole communities, and what kind of treatment you can use to prevent the apparition later on. So not to put things when the disease is present, but you can know that the disease can appear before, and you can take some measure for that.
Very interesting, the approach that there is not only one microorganism involved, but many. That's very interesting, especially if now we're able to see the whole community. But going deeper in what you say about how the microbiome, the terroir is changing because of the climate change, how could this affect to the flavors of the wine, to the fermentation process, in the cellar?
Well, you can maybe think about the fact that changing the microbial community can lead to a situation in which you are missing some of the bacteria that before were affecting like the taste of the flavor of the wine along the fermentation. Or at the other side, you can change what was the relative abundance between those microbes like in two different moments along the year. And this can also lead to like a change in the final composition. I think like so we know that like we can find like many different species and also many different species that are related with the fermentation. We can find them like in the vineyards, and in somehow especially when we are talking about spontaneous fermentation, so the grapes that are being harvested and then directly fermented without like inoculated with a commercial strain or something like this, then here you can see, especially in the early stage of the fermentation, a huge impact of what comes from the field, and also of course from what comes from the winery. But if we think that the microbiota inside the winery is relatively stable, then we can maybe check what is going on in the field, and this can be affected by so many factors that are also environmental, like the temperature, the moisture, and in general the weather. And in this, how you can see maybe a difference in the final like in the chemical and composition of the wine.
Yeah.
I think I agree with Alex. For example, in our data, when we analyze fermentative samples in the beginning stage, there are some wineries to use, for example, or add dry ice to the wine to decrease the temperature of the fermenter and improve to extract more specific thiols or compounds, no? Right. When we analyze the microbiome in this stage, we detect that the different non-Saccharomyces yeasts are with activity, and when this change of temperature in that specific moment improve that species can start to growing, but decrease the activity of Saccharomyces. For it, these type of practices we can improve the flavors in the wine. this is only one example. Other example, for example, related with our data, when we analyze the surface of the grapes, we can find more than 800 different species, bacteria and yeast, and 50% of that species are in samples related with the fermentation, coming from the same grapes. It's incredible because there are a lot of different players in the wine, and we only now know 1, 2, 10 of them. And we can detect too, for example, when the climate change, or if we analyze the same grape in the same parcel year per year, we have data from 3 years. We can detect change in the abundance of Saccharomyces. And last year we started an experiment, and we analyzed the grapes previous to the Envero. Is the correct word, Envero? Yeah, okay. Previously to the Envero, and from the Envero to the harvest. And we can detect how specific species related with the fermentation process as Saccharomyces grow to the best moment to collect the grape. There are a microbial maturity in the grapes too, related with and we can relate with the flavors.
Okay, so yeah, I also found that is extremely actually interesting, the fact that you can detect so many species on we can actually detect so many species on this. Oh yeah. And at the other side, we also known from research that has been done in the past that Saccharomyces on grape is extremely rare, but then it takes advantage very fast during the first stage of the fermentation. The fact that you maybe can slow down the rise of a Saccharomyces, then it can improve the complexity of the wine that comes from many other different small actors that can act during the early stage of the fermentation. Yeah, and I think also this is very important when you get spontaneous fermentation. Particularly, I follow only spontaneous fermentation because really I want to do this. So I want to track the microbial community from the very beginning of the from the field, basically to the final product. So when it's going to the bottle.
I really like that you are not looking at the microorganisms just for the, let's say, negative part, but also how these microorganisms can promote and improve, well, the whole process, the whole workflow. Coming back to one thing that you mentioned before, and in the previous session, we hear the concept of precision enology. So right now, if I understand properly, the capacity of the winemakers to modulate the process is quite limited, because if we look at all the commercial yeast, that is more or less the same all around. Are we moving more in really precision enology, understanding more like we will choose with the species and then find that aroma profile that probably winemakers are pursuing at the beginning of the process?
Yeah, so the more we get information about the physiology of the different yeast and the different bacteria, then we can also think about making a commercial inoculum, commercial starter that are a blend or a mix of different strains in order to try to add a little bit more complexity to the industrial wine. And I think this is interesting when it is done in very controlled conditions. So when we know very well which kind of metabolites can be produced using this way. But yeah, I think it will, I mean, now there is a very big difference between what is produced with a commercial strain and what you can find with in terms of reproducibility reproducibility of the product along the different years. So if we can find a way to produce a starter that is extremely more, let's say, more complex in the profile of aromatic compounds that it produced during the fermentation, then it would be probably would take place relatively fast between the winemakers and the different wineries, because I guess it's something that they are looking for, because consumers want a particular wine, a unique wine. They want to feel like from one part they want to feel the same taste along different years. From the other, they want to feel also like a rich taste in all the different wine they are gonna try. So in this way, I guess it's important to make experiment and research in order to understand better the physiology and the metabolic profiling made by this new yeast or bacteria that are being isolated. Yeah.
So knowing that is really interesting, the question then is which kind of techniques do we currently have, Alberto, to explore all the possibilities that knowing the microbiome can give us? I mean, there could be culturing, there could be, please.
The most traditional techniques is culture, isolate the different species and culture the species, but this has a specific problem. We don't detect all the microbiome community. We are using a metagenomics approach. We only need to collect a sample, isolate the DNA, and then use a specific gene markers associated with taxonomy, or we can use or read all the DNA in the sample directly without culture. This is very important because with this we can know new species too. But the problem right now is a database problem because the technology is moving fast. We now can apply technology that when the people sequencing the whole genome of the humans, the technology is now more or decrease the price. Is very accessible for all the people, but the problem is the database. If we can move from a specific species level taxonomy from a strain level taxonomy, we need reference. And at this moment, it's very difficult to find high quality reference. Probably important groups of research like you can apply for new projects related with this. Because, well, for example, a specific question a lot of winemakers ask me is, okay, when I start a spontaneous or inoculated fermentation, is my yeast or is other of the yeast in the cellar, or is the yeast from the last year, or is an hybrid? These kind of questions we can solve with the genomic technologies, but we need to improve the quality of the database and increase the number of genomes in the database. Sorry. For the other hand, only with the genome or genome data, only use genome data no is the best. Because we can collect a lot of DNA, but we need to compare the DNA with something. We need to correlate the DNA with something. For example, in Biome Makers, we use 119 different packages of data we can collect associated with 8 samples. For example, related with soil sample, we can collect the location of the sample. Associated with the location of the sample, we can collect all the weather information related with the parcel in a year. Temperature, humidity. I think it's crazy the number of data we can collect associated with the location. And the next step is correlate all this information with the microbial profile we find in soil or in grapes. We need to build a model system similar to the weather prediction system to create this information actionable to the users. For example, prevent disease or anticipate the flavors of the wine according with the specific non-Saccharomyces and Saccharomyces yeast we can find in the surface of the grape.
Yeah, and another thing that I was like talking about like this is like this is a technique that has been like pointed as biased in somehow, but then is not more biased than the techniques that were used before. Like before also there were a huge bias when we are talking about the cultural dependent analysis. Then we have a big bias because we only can detect, I don't know, 5% when we are lucky about the microbial community that is there. So the other 95% we cannot see them. And also we don't know like if this 5% is also the most abundant, it's just the one that is cultivable. With the other like molecular techniques like the previous like the one that we were using before the sequencer, then also there was like this kind of issue. And now of course we still have some bias, but this doesn't necessarily mean that we cannot see anything. I mean, it is always like a matter of seeing different point of view the microbial community, and in this way at least we can get access to a very wide part of the picture, while before we could only see some spot of it. So I guess of course we the future will give us better technologies, and I hope we will, but at least now we can still find some correlation if we try to keep stable the technology and apply it in the same way to all the samples. So in order to get the same error from all the different samples, in this way we can then compare them and see the answer to some of the questions that we already have.
So Stephane, you are not talking a lot, so let's come back to a very interesting point that it's clear that there is an impact by knowing the microbiome, what is there, which kind of species. So thinking about how to provide value out of this information, let's think about sustainability, both from the environmental point of view and from the productive point of view. How can we make the vineyard production more sustainable?
We should make more organic approach first. And but you know, when we make organic approach in the vineyard, so we don't use any chemical product, but we also we will reduce pathogen with some biocultural agent, but we also increase the biodiversity of the soil microflora, of the good microbiome. Because when we use normally some chemical products, we have direct effect on the pathogens, but we have also indirect effect on the good microbiota associated with grapevine plants. And so when we do more organic vineyard, we have a better biodiversity. We can have, of course, apparition of pathogens, but we have a better equilibrium of the microflora, not impacted by chemical products from outside, but only from environmental condition and cultural practice. And for example, you know, when you go to the vineyard, even when they are managed under organic condition now. Last time I was analyzing some genome of bacteria, and you know what I found? I found these bacteria, they were resistant to arsenite and copper to a high level, because they make adaptation to the vineyard. And if we use after less chemical products, we will have a microflora coming back to the place and make more diversity of microbe. And we know that chemical products also influence the biodiversity of microbes, and that the organic approach also impacts the biodiversity of microbes associated with grapevine plants. But we need to protect our vine, we need to have less disease, but we need also to take account that there is some good part of the microbiome associated with grapevine plants that help for vine health and also for winemaking process.
So at the end, the conclusion is let's be careful with what we throw into the field, because it could be also decreasing our productivity, isn't it? Okay, good. And then by having this information, how can we also value that from the, because at the end, the final product coming from the vineyard is going to be the wine. So how can we use that? Alberto.
With the economical perspective of use more sustainable, be more sustainability in the vineyards. From my perspective, there are a lot of viticultures working on that. For example, that data from Spain, there are more than 500,000 people working on the vineyards. There are more than 1 million of hectares in Spain for vineyards. It's a very economical perspective. If you compare or if you calculate the losses when, for example, the people lost a complete vineyard, are crazy, because not is the loss to start to replant the parcel. It's the losses to you can produce wine in 5 years. It's more than 150,000 euros in these 5 years. It's an economical problem. Of course, I think the vineyards is a culture, or the wine is a culture in the Mediterranean countries. And we need to prevent these problems. We need to prevent the problems with other perspectives. Not a reactive perspective, if not a preventive perspective. If we change our mind in that field, in that direction, probably we can change this, and probably we can stop to lose a lot of money with this kind of problems.
You want to add?
Yeah, well, I mean, about the sustainability, yes, of course. I mean, I'm mostly like concerned about like the environmental issue, and of course, I mean, it's important to try at least to reduce the use of like any chemical but I guess the final aim is to be able to produce like good wine in enough amount, and so I'm totally open to the use of like any biocontrol agent as soon as it will demonstrate to be, of course, like working. And I guess like you can either hope in protecting the grapes when they are like on the plants, and also like trying to influence the fermentation. And the best approach would be like to join these two like effect in one single operation. That would be like inoculate with a strain with a specific bacteria or yeast that can occupy the niche on the grape skin, on the surface of the skin, in order to prevent the attachment by any pathogens, and also like that you can find the same bacteria inside the fermentation tank, and is also like able to produce like some secondary metabolites that makes your wine better. So I guess from the point of view of the sustainability, I mean, economical, of course, environmental also, and in this case it will probably be the best choice, like to be able to join these two effects in once.
I wanted to say also things we need to protect our vineyard, and we need to protect also the good microbiome associated with the vineyard. Because if we have a problem in the good microbiome, we will have more pathogens, and we will have more disease. And after, we need to find curative solution. But there is good part of the microbiome. There is the good, the bad, and the uglies. And the producer of mycotoxin, for example, for me, are the ugly microbes. But we can protect the good part of the microbiome by knowing first what kind of part of the microbiome I have in the soil, in the plants, in the berries, in the vine. How can I increase the good part of the microbiome and reduce the bad parts, the pathogens? And if we know before, we can make preventive solution, use inorganic compounds or organic compounds, depending of the vineyard approach. And we know that the plant is not alone, and we need to protect also the good microbes associated with the plants, because otherwise we can get a lot of problems, like when we have done before with using chemical products. It was good for the plants, but it was not good for the human health. And it's the same for the good microbiome of the plants. And some of these microbes, when we put some biocontrol agent on the vineyard, you know that grapevine plants, there is some plants, they are more protected than the other plant in the same vineyard against some disease, because they interact with some good microbes already in the soil, inside tissue. And when we know which kind of microbiome is associated with a terroir, for example, it gives the characteristic of the terroir as a fingerprint, but also it gives indication what is the risk after to have such kind of pathogens, for example, or also what good part of the microbiome I have associated with my vineyard. Because we have not only pathogen associated with grapevine plants, we have also a very big proportion of good bacteria and good fungi helping the plant's health and the plant nutrient acquisition.